Okay. Yeah, thank you. So I just want to welcome everyone here. Happy New Year. Welcome back. For those of you who have had some holiday time, I hope you've had some holiday time. This is the next installment of the active post-acute sequelae of COVID-19 seminar series. We've had a number prior, and they are all available to be seen on the website. So, and I think that website is in the chat. If it's not, it will be put into the chat again. My name is Joe Manetsky. I'm the Vice President of Research Partnerships at FNIH, and I've been helping lead and guide the preclinical group within Active. And what you see here are what we've talked about. So the top 5 are those talks that you can find on the website. We're going to have Dr. Reiser here talk today about COVID and the kidneys. And so that's always interesting. We will be having actually 2 in the next couple of weeks on pediatric PASC. So they will be coming up. And the plan here is that these seminars will be put into a white paper or something and probably on the website for continued use and reference. Just want to remind everyone logistically that presentations will be recorded for transcription processes, but these they will be available. The actual recordings will be available in the future. The transcripts from the seminars will be made available in the future. The attendees are asked to mute during presentations, and we will do some unmuting when we get to the Q&A, which we always seems to be the fun part of another fun part of the meeting. The Q&A actually can be facilitated more easily by the host if you use the Q&A feature. So Zoom's got one. Let's use it, and if you put your questions in there, then we can make sure that they get to the speaker and addressed. So please use that. And you'll notice the boxes at the bottom of your screen, whichever screen you're looking at, should be able to be seen to identify the Q&A and the chat features. Okay, and with that in mind, I want to introduce my hosts, my extremely capable and now good friends hosts, Dr. Jay Rappaport and Dr. Josh Fessel. Both of them have been working on this program and with the preclinical group since its beginning in April of 2020, which seems like forever ago, but that's where we are. Jay is, as most of you know, is the director of the Tulane National Primate Center and is the chief academic officer and professor of microbiology and immunology at Tulane. And Josh is not at NHLBI anymore. I think he's at NCAT. So we need to make a change to this slide, but he can explain where he is. But at any rate, so no, he's a program officer at NCATS, both of which have been leading these and hosting these seminars, and allows me to actually just listen and enjoy the science. So with that, I am going to turn it over to Jay and let him introduce our speaker today. Thank you.
Well, thanks very much, Joe. It's really a great pleasure to introduce today Dr. Jochen Reiser as our distinguished speaker for our continued series on PASC. Dr. Reiser is a world-renowned leader in research and also healthcare. He received his MD and PhD from the Ruprecht Karls University in Heidelberg, Germany. He did his residency in internal medicine at Albert Einstein College of Medicine, and he then did his fellowship in nephrology at Mass General Hospital, Brigham and Women's Hospital, and Harvard Medical School. He's currently the Ralph C. Brown Professor and Chairman of Medicine at Rush University, and also at Rush, he's Chief Innovation Officer. In 2017, he became a member of Germany's National Academy of Science. And in addition to other awards, he also received the Halo Hero in Medicine Award in 2019 for his work improving the lives of patients and contributing to human health. He's co-founder of a biopharma company, Walden Biosciences, and some of the history of his work, especially the work that's pertinent here, was described in a science paper in 2019 called Omen in Blood, which described the risk of patients with focal segmental glomerulosclerosis, or FSGS, and patients who needed renal transplants. The same thing, tended to happen in the transplanted kidneys. And Dr. Reiser figured out that this was due to something in the blood, something he discovered, which was the secretive form of the urokinase plasminogen activator receptor, or suPAR. And this has been a really important discovery. It sparked numerous investigations and now a target for therapeutic interventions and many novel clinical approaches. And very pleased to welcome Dr. Reiser to introduce a seminar on PASC, COVID, and the kidney, a suPAR example.
Wonderful. Let me share the screen. Okay, I think you should be able to see that. Can you? Yes.
Yes.
Wonderful. First of all, I'm truly humbled to be invited to this distinguished panel. And it's an honor, and it's my first talk in 2022, so I'm particularly excited to start with a talk like this. I've been working on kidney disease mechanism for my entire career. It started as an MD PhD student already, and we defined a role for an immunological factor that comes from the bone marrow, which is called suPAR, and I'm going to guide you through that work. And when COVID hit, we were at that time already intrigued and figured it got to have something to do with this because it's so strongly stimulated in its expression by RNA viruses. And truly, it has a very important role. How this plays out for PASC is still under heavy investigation, and there's a lot forthcoming. And so I'm going to guide you today through that entire work, how immunological factors, in particular suPAR, are a strong risk factor, and how they work in kidney disease and also in COVID, acute, and most likely, and I say most likely because we don't have the data yet, in PASC. I do have a conflict of interest that I'd like to point out. Dr. Rappaport already mentioned it. It's Walden Biosciences. It targets suPAR and other factors. So please keep that in mind. Why do we need to talk about kidney disease in particular? And I think this slide really says it all. We have an enormous medical condition, a very sort of silent but very prevalent condition is loss of protein in the urine, or called proteinuria. At least 500 million people have that particular sign of early kidney disease, and it's itself a risk factor for both extrarenal and renal disease and mortality. It's a very, very expensive condition in internal medicine. The annual cost of treating kidney disease, in particular end-stage renal disease in the United States, is over $32 billion. And currently, we have almost 800,000 people in America that are on end-stage renal disease. So this is a very growing problem. And we can't permanently fix it. So even with a transplant, we see recurrence of diseases or loss of transplant function after years, and then we have to deal with it again. And the annual costs to treat kidney disease are more than 1/4 of Medicare's expenditure. And this will increase in years to come. And with COVID and post-COVID, we will have an increasing amount of people with chronic kidney diseases or accelerated chronic kidney disease. Drug development, and please note this slide is from 2012, has historically been lacking for kidney disease when you compare it to other diseases of similar healthcare burden, such as heart disease, cancer, HIV. We had very scarce development, and that is because the pathobiology of kidney disease is very complex. We have 36 or 37 different cell types in the kidney. Compare this to a heart. It is probably a lot more complicated in the interaction of these different cells and how they work. And with that, it lacks drug development. That has changed somehow in the last 10 years. We have now more companies, preclinical and clinical, that are in this space. And some of it was driven by a more appreciation of the healthcare problem, but also of some regulatory changes, for example, using surrogate endpoints in trials, such as proteinuria reduction instead of long-term preservation of renal function, at least in orphan diseases. So that has gotten better. I like to simplify kidney diseases in 3 different categories. One on the left side is the physiological component of the perfusion. We all know that blood pressure is very important for kidney function. And with that, we also can appreciate that reductions in blood pressure is beneficial. So ACE inhibitors, angiotensin receptor blockers, the newly approved sodium glucose cotransporters inhibitors, they all optimize the physiology systemically and in the kidney, and they have benefit. They can slow down chronic kidney diseases. They can treat them definitively, but slow them down. And then there's the second category, which I like to summarize with structure or genes. We know there are a lot of mutations in the kidney, and they are risk factors. And because of these risk factors, kidneys fail earlier. And hopefully one day we can fix them with CRISPR. It seems to be becoming more likely that this is the case. Or we can modify the pathways with drugs that are disturbed from a gene mutation. And then there's the third category. Now I got interested in that right from the beginning of my career, because I have a soft spot for immunology and inflammation. I thought this is so important, especially as we get older. And if you think about one of the major drugs that we're using in kidney disease, it's steroids. And steroids, we give steroids for nearly everything in nephrology, and they either work or they don't, but often they do work until there are side effects that precludes further usage. So there's always this idea that if we could harness the immunology better, we might have better treatments. And the soluble urokinase receptor is a molecule that comes from the innate immune system, and we'll come to that. So here you see again how the immune system is really surrounding the kidneys heavily. Some of the key cells in the kidney are the podocytes, which are responsible if protein is spilled into the urines. These cells fail, and they are the key cells of chronic kidney disease. And inflammation can be affecting podocytes on multiple levels, directly with immune cells that attacks the kidney cells, or through other renal cells that are being attacked by the immune system, or through cytokines, complements, and other circulating factors that run through the bloodstream into the kidney. Now, when we look at the podocyte, we have to recognize the podocyte is part of a unit we call the filtration unit. Here you see these foot processes that sit on the glomerular capillary. In between is the thickest membrane of the human body, the glomerular basement membrane. And these 3 elements are the filtration barrier. And we can look at this barrier with electron microscopy, but now with newer techniques, we have learned that this is not a static structure that's either there or broken. This is a highly dynamic structure, similar to dendritic spines. This structure is in motion. These foot processes are dynamic, they can move. And disturbances in these dynamic assemblies are underlying disturbances in the kidney filter and also early kidney diseases. Here you see an electron micrograph now of one of those glomeruli. We're looking from the urinary space onto glomerular capillaries that are wrapped by these podocytes. Here, one cell is highlighted. See this intricate structure of these filtration slits. And as I said, these structures are not static. They're highly dynamic. They're going in and out, and based on blood pressure and filtrate, there can be modifications. We have about 1,000 podocytes per glomerulum, and we can tolerate a loss of about 20% of the cells. And once we go below that, then this glomerulum will form a scar. And with 2 million glomeruli in both kidneys, we have about 1 million glomeruli at capacity before the kidney function then will go sour. So here you see a section through the filtration barrier, fenestrated endothelial cells, GBM, podocytes, filtration in this direction. Now, the earliest failure of podocytes is called effacement. And here you see the intricate structure where these foot processes interact with each other. They are being pulled back and they fuse. And when that fusion occurs, then protein can transfer from the blood vessel into the urinary space. Even though this looks so more tightened up and sealed up, it's actually much more porous, and more material can transfer because the foot processes don't work properly anymore. If this is not fixed, if this is not recovered, then these podocytes undergo more changes, including detachment. So they will start to fall off in part from the capillary, and then larger pores and holes are being developed, and then a sequelae of histological changes occur, cell body attenuation, pseudocyst formation, and so on and so forth. And that leads to what we call a scar. And once the glomerulum is scarred, then the tubulus that is underneath will also be scarred. And here's an example of human diabetic kidney disease. It's a very stereotypical progression of chronic kidney disease that we see in many, regardless of what the cause was. You can see the same in a lupus nephritis. Of course, you have more inflammatory component, but you see the glomerulum scarring and the tubulus scarring in a similar fashion. So how can we study effacement, something that is by electron microscope? And we brought this in vitro with highly differentiated podocyte cells 20 years ago. And these cells have been optimized by many others. But this was actually part of my MD PhD thesis. I was the key driver, so to speak, in the laboratory, of course, with my PIs at that time, that developed these podocyte cell cultures that are differentiated. When one puts nephrotoxins on these cells, one can see the simplification of the actin cytoskeleton, and there's an increase in motility. And this increase in motility can be measured. And if someone deletes key regulators of motility, such as the urokinase receptor, then this motility goes down or is stabilized. So those are just some very rough assays, how we think about the beginning of effacement and how this is regulated at a cellular level. Here you see another image where you see the black spots representing podocyte cells that migrate under normal conditions over 24 hours, or under nephrotoxic conditions. There's much more migration, and that is a surrogate for a failing podocyte. Now, these podocytes are attached with their foot processes to the GBM with integrins. And that is, again, nothing too special. Many cells have integrins, of course, they have to attach and adhere. But what's interesting with the podocyte is there are static integrins that are more providing the function of adhesion, and there are regulatory integrins such as alpha V, beta 3, that are largely expressed, but they are inactive in their stage, and they can be activated by something like suPAR or uPAR. And I'll come to that. I'll show you some experiments. Here you see the foot processes. This is upside down on purpose because it fits better the depiction here. So we have these two integrin systems, alpha V beta 3, which is largely expressed, but it's inactive. So it's a perfect regulator because it can be activated by something. And then there's the other large group, alpha 3 beta 1. If they're being deleted genetically, then there are no foot processes to be formed right from the beginning, but they're always active, though they're not good regulators. So that's it's a big difference between these two integrins. Again, one can delete alpha V beta 3 from podocytes. There's no phenotype, but then these podocytes are resistant to more motility, and because of that, they're resistant to the onset of disease. So keep that in mind, because this will become important. So what we found is years ago, and we've built on that, is series of papers showing that UPAR on the cells or suPAR from the bloodstream can bind to the beta 3 integrin and turn this integrin in the active configuration. And that activation leads to an increase in motility, to activation of cellular programs, small GTPases, and so forth, that leads to the effacement process, and with that to the beginning of the disease. And that was summarized, as Jay has mentioned in the introduction, in this 2018 paper. It's a fun read, actually. It's 5 pages about these two identical twins who I know very well. They have been my patients, and we're still very much in touch, and they like to go public with this, where the identical twin was bitten by a brown recluse spider, and within a few days developed severe effacement, nephrotic syndrome, developed kidney disease, lost his kidneys, and got a transplantation from his identical twin brother, and then shortly after transplant recurred within a few days and had again massive proteinuria and lost his kidney again. When we measured suPAR, the patient had very high suPAR levels, whereas the twin brother had very low suPAR level, sort of like hinting at the idea that something immunologically has gone off that's driving that process. And we know now that suPAR comes mainly from the bone marrow, from immature myeloid cells that get stimulated by viruses or toxins or other events, such as SARS-CoV-2. The levels of suPAR rise in the blood, go through the capillaries into the kidney, and bind to these integrins and turns on activation in these cells, and that leads to proteinuria kidney disease and other manifestations, which we have seen in acute SARS-CoV-2, and which we are also starting to see longer term in patients that have persistent higher levels of suPAR. We'll come to that. So what is UPAR and suPAR? It's a very, very interesting molecule. It's a 60 kilodalton glycoprotein. It's fundamentally a toxin. It's fundamentally something like bucandin. It belongs to the group of 3-finger toxin. And you can see here bucandin with these 3 fingers. The same structure is essentially there in the UPAR-suPAR family. And that's why it is also expressed in nanogram per milliliter levels. It's a very low expression in humans. In healthy humans, it's around 2 nanograms. But I think it's being used in the innate immune system as a messenger to signal to other organs, for example, the lung and the kidney, and we'll come to that. It has 3 domains, a D1, D2, D3 domain. The D3 domain is linked with a GPI anchor to the cell that can be cleaved off. And then there can be an additional proteolytic cleavage between the D1 and the D2 region, cleaving off a D1 domain. So this is the typical form of UPAR, and when the D3 is cleaved away from the membrane, and we call it suPAR. It's at baseline, UPAR expression is low and very specific. There's a high expression on the RNA level in the bone marrow and immune system because it's part of the innate immune cascade. And there's also some protein expression at baseline. Other organs have very low expression other than the lung. In the lung, interestingly, it's in the bronchial tree, in some of the stem-like cells down the bronchial tree. And that's why we also think that viruses that get inhaled and bacteria, that get inhaled are particularly stimulating expression, because there's some sensing going on that we don't fully understand yet. And as I explained earlier, if the cleavage occurs between the D3 domain and the plasma membrane on the GPI anchor, then the full-length suPAR is released, and that's full-length suPAR. And then this suPAR can be further cleaved in a D1 domain and a D2, D3 domain. Now, suPAR can be measured with ELISA systems. There are now multiple sandwich ELISA systems available. Two have been very well utilized. One is the Quantikine assay in the US and the suPAR Virogates assay. I have nothing to do with any of these tests, but we know now that these ELISA assays, they correlate fairly well with each other. But now, as the biology has gotten so much more interest, people are using also proteomic platforms to measure suPAR and uPAR as part of a multi-marker panel, so to speak. And we have to say, and there's publications now coming out, that the correlation between the ELISA testing and the proteomic testing systems is very poor. So as per now, with respect to kidney outcome prediction and cardiovascular outcome prediction, I would recommend, and the field recommends, to stick with the ELISA platforms until we have additional assays available. Here are the normal levels in women. The levels are around 2.4 nanogram per milliliter. They're slightly higher than in healthy men, which is around 2.2 nanogram per milliliter. But the threshold for disease is higher in women than in men. So this is balanced out, so to speak. And one of our initial discovery was that people with a certain type of acute proteinuria kidney disease, it's pretty aggressive, FSGS, before and after transplant, which we call recurrence, have higher levels of suPAR in the blood compared to patients that have other kidney diseases or have no kidney diseases. And that persisted after transplant. So the one patients that had, again, recurrence of disease have still elevated levels, whereas the one that had settled down with their kidney function, the levels of suPAR were also lower. And this is work from 2 years ago, sort of like a meta-analysis, and this is an additional assay where people compare the different testing system. And you can see that the Virogates ELISA separates FSGS better from other glomerulonephritis, other kidney diseases, than the R&D assay, where the window is just narrow, and we need a larger sample size to see the differences. So right now, the recommendation is to use the Virogates assay. Again, we might have more assays or better assays available in the future. So as it stands, suPAR comes from the bone marrow after stimulation of infection with bacteria and viruses or other types of inflammation, is released from these GIA1 low immature myeloid cells. We published very nice work in Nature Medicine in 2017 together with David Skadden at the Harvard Stem Cell Institute, could show that these cells expand, produce suPAR, and then suPAR comes to the kidney, activates these integrins, and then there's proteinuria. And this is really a wonderful immunological cascade, how the immune system can regulate kidney functions. In the meantime, we also have found some endogenous defense mechanisms. One is the ICOS ligand system. I don't have time today to talk about it, but we have published quite extensively about this, but this system can get easily overwhelmed by a too high or too persistent suPAR level. So when we tried to figure out where suPAR is coming from, we did a very interesting set of experiments in this Nature Medicine paper Unsel Ham did. In the meantime, also NIH funded and has an independent laboratory. We went to patients with recurrent FSGS who have high suPAR level in the blood, who had the disease come back after transplant. We took their PBMCs and infused them into NSG immunodeficient mice. And interestingly, what we found is that after about 10 weeks, these mice that were humanized from these PBMCs from the patients developed proteinuria kidney disease and massive effacement of their kidneys. Whereas the other mice that received PBMCs from healthy people had, after 10 weeks, no proteinuria and normal podocyte foot processes. And interestingly, we saw the immature myeloid cell expansion in these humanized mice. They expressed GIA1 low. We saw the increased uPAR expression on these bone marrow myeloid cells, and we saw a rise of suPAR in blood and urine. So we were able to transfer the immunological program from patients into these NSG mice. And we're using them today for drug development, as well as for studies to better understand what happens after an infection with a virus, for example. Other work here goes back to 2008. This was one of our first paper helping us to understand the role of suPAR, where we took a UPAR-suPAR knockout mouse, which has normal podocyte foot processes. As you can see, the stimulation with LPS, which is a nephrotoxin, leaves the foot processes intact because the activation of the integrins cannot occur in the absence of UPAR or suPAR. However, if one re-expresses the UPAR in podocytes genetically by bringing it back into the cells with a podocyte-specific promoter, and then give these animals LPS, then the nephrotoxin is able to activate UPAR expression. The UPAR starts to activate the integrins, and there's foot process effacement. So this was an important finding that highlights the importance of the activation system that we proposed is happening in the onset of the disease. Now, also when we express the UPAR in glomerular endothelial cells with an ICAM2 promoter, this was still not allowing for the activation of the integrin in podocytes. So the UPAR-suPAR action has to occur in the podocyte integrin system and not in the integrin system on the cell next door, so to speak. Now, here's another example where we expressed a suPAR in mice that is the E134A that's incapable of binding to integrins versus this version, suPAR12 is. And one can appreciate that there's heavy effacement that occurs in short order, whereas the UPAR that's genetically transferred, that's incapable of binding to the integrin because it's mutated in the integrin binding site, the foot processes are left intact. In the meantime, we also created transgenic mice, just very standard, and watched these mice over a year, year and a half. And you can see here in the isoform 1, this is the suPAR that has 3 domains, D1, D2, D3. After 2 months of high suPAR, some of the mice, not all, typical for a risk factor, less than a third, start to develop proteinuria and larger glomeruli, whereas the isoform 2, which is a very strong integrin binder, 80% of mice become very proteinuric, and mice start to have sclerosed and scarred glomeruli much earlier. And you can see here, I'm going a little fast because I want to come to the human studies. You can see the difference between suPAR 1 and 2 is on domain 2, there is a change in sequence, and domain 3 is missing completely. We actually 10 years ago were puzzled by that because we had such a nice phenotype in mice, but structurally we didn't think it made too much sense, and we wanted to understand more about it. So in collaboration with the Harvard Medical School Structural Biology Core, we were able to purify this shorter version of suPAR, suPAR 1-2, and we could derive a structure that let us appreciate that it's a dimer, so that the abrogated D2 domain is fused to another D2 domain that's shorter and a D1 domain. So it's a dimer. And that is probably why this particular suPAR is so strongly activating integrins that the disease in mice becomes very prominent very fast, and the kidneys fail very rapidly. And we characterize these mice, they get heavy proteinuria, they get renal failure, they really have nephrotic syndrome, so to speak. And they scar, and they have heavy effacement in short order. But interestingly, when we cross these mice with integrin knockout, so when we don't give them the integrin to activate, then the phenotype can be rescued. Here are 3 different types of integrin beta-3 knockouts. And then also the phenotypes are much better. The glomerular capillary are opening up again. There's less scar as compared to mice that have a competent integrin system where the glomeruli are being scarred. I want to show you on this SPR how suPAR acts at the integrins. And here we are comparing a normal suPAR, so to speak, the 3-domain, versus this very aggressive 2-domain. And I'll play this a couple of times. Here's the 2-domain, here's the 3-domain. They both interact with the integrin in the nanomolar affinity range, so very tight binding. However, the abrogated form comes on and off very rapidly, and the off curve is essentially not there. It's such a sharp drop, whereas the other, sort of like the 3-domain suPAR, comes on much slower, stays on longer, and comes off slower, giving the cell the ability to react and counterbalance. So the not only the binding of suPAR to the integrin is important, also the pattern of on-off and how long it persists at the level of integrins, something we're learning more and more of. In genetic studies, we then see that in these mice that overexpress in the kidneys this short version of suPAR, we see upregulation of integrins. That's not a good thing. And here is a study from St. Louis, from WashU, single-cell RNA-seq studies in humans with early diabetic kidney disease. There's a strong upregulation of integrin beta-3 in podocytes of people who are about to have diabetic kidney disease. And just showing you some of the relevance. Now think of this together with a high suPAR, upregulation of the receptor in the kidney. That can be a perfect storm scenario. Now, this is just a snapshot now of the biology of suPAR and what it does at the kidney. Now, I would like to show you some epidemiological studies that really put this in a much larger context than any of us, certainly myself, would have had anticipated. So in 2015, we said, okay, let's now not think so much about the molecular mechanism, but let's take a large cohort of people and measure suPAR. And here we took the Emory Biobank, about 3,500 patients that come to the emergency room with chest pain. So this could be an anxiety attack or it could be a heart attack. It could be anything. And when we looked at that in these patients, we saw that suPAR was the strongest independent predictor of decline of kidney function in patients that already had kidney disease, but more excitingly, perhaps, in patients that had absolutely no kidney disease, a suPAR level in this cohort of higher than 4,020 picogram per milliliter led almost to a 37–38% prediction of future chronic kidney disease within 5 years. And so this was a very important finding. And of course, we wanted to know, could this be just a reflection of already pre-existing kidney disease that the suPAR is high? But it's not. While suPAR is correlated to renal function, at a kidney function of 90% or more, so 90 mils per minute, there was no more correlation. Overall, the correlation of suPAR with kidney function is very, very small, even in lower kidney function ranges. When we looked at other markers that are associated with decline of cardiovascular function, for example, HSCRP, other inflammatory marker, HSP70, troponin I, and fibrogen degradation products, none of these markers predicted decline in kidney function independently. Only suPAR was associated with a decline in eGFR, but compounding those markers together, the risk prediction could be even further improved. So that's also a very interesting finding, but pointing to suPAR as having a special role in the connection between the immune system and the kidney. We also found that the higher the plasma suPAR, the more likely patients had proteinuria, which is associated obviously with the activation of the integrins that I explained to you at the beginning of my talk. Now, there was validation. Here's a big study from Sweden in the Malmo cancer diet, where 3,000 some patients were examined over 20 some years, and just the suPAR levels were taken into quartiles, and then the annual loss of kidney function was charted. And what you can see even in entirely healthy people, the higher the plasma suPAR, the more there is loss of annual kidney function. And over 15 years or more, this resulted in more hospitalizations and more kidney diseases. So even the patients on the quartile 2 already after 15 years had way more kidney problems and were also hospitalized for kidney diseases. This was validated in another study in China. I don't even need to go into great detail. The higher the suPAR level, the more likely this patient progressed to end-stage renal disease. Here in African Americans with an underlying genetic mutation, APOL1, which is a risk factor for kidney disease, what we could show is, and I think this is very important finding, even in the high-risk genotype, if the suPAR level is not high, but normal, which is around 2000, then the annual loss of renal function is almost if the patients wouldn't have a genetic mutation. And which really highlights the role of the immunological compound on top of other existing risk factors. And that was essentially validated in another large cohort out of the University of Washington. And also it's the same was true in sickle cell disease. So sickle cell patients who have the trait are much more likely to progress to kidney disease in the setting of a high suPAR than in the setting of a low suPAR. Similar finding in cystic kidney disease. This is autosomal dominant kidney disease. I mean, very, very striking. So essentially what we learned, and then I can come over to COVID more, how this fits in there. Patients who have low suPAR and normal kidney function have a low risk of future chronic kidney disease. Patients who have normal kidneys but high suPAR, for example, in response to a COVID-19 infection or in a response to a persistent elevation of suPAR under long COVID, have a much higher risk to develop chronic kidney disease. Patients that already have existing kidney disease are much better off with a low plasma suPAR because the immunological risk factor is low and there's less progression. Patients with already existing kidney disease, let's make this a lupus nephritis or genetic kidney disease, now with a high plasma suPAR, for example, as a consequence of COVID infection, are under much more risk for future progression. Now, with that in mind, we thought, could it be that acute kidney injury that occurs in the hospital typically, that's a very rapid effect where kidneys fail in response to toxins or low blood pressure or sepsis. Could patients who have a higher suPAR be already but normal kidney function already be at a risk for AKI? Because right now, the way we stratify AKI risk is by pre-existing chronic kidney disease with the typical markers like creatinine or plasma markers that we know. For example, urine EGF has been shown a very early CKD marker, but already in CKD. So these patients have already kidney disease, and then the EGF will give us further insights into how aggressive it might be and into how much this might be a risk for AKI. So we thought, okay, let's go even earlier. Let's say we just have patients with a higher suPAR, but no kidney disease yet. Is that already a risk for AKI? So we looked at that and we were surprised again. We took patients, in this case, 3,827 patients, all comers, who were undergoing contrast dye infusion for coronary procedures. Young people, old people, pre-existing conditions, no pre-existing conditions. And no matter how we modeled it in this analysis, the higher the plasma suPAR before the procedure, the more likely the patients develop AKI in hospital. And you can see here in this forest plot that this was independent of pre-existing chronic kidney disease, diabetes, yes, no, very striking findings that we all were really puzzled by. Validated in an independent cohort here in the Brigham and Women's Hospital, a 250 patient who undergo high-risk cardiac surgery, cabbages, same thing, plasma suPAR levels before the operation predicted the development of AKI post-procedural. Sepsis, 692 patients, also from the Brigham, sepsis, AKI, similar pattern. And in an additional paper last year, for the first time, very important paper, patients acutely admitted to a hospital for COVID-19, if the suPAR levels went above 6.86, then these patients had an enormous odds ratio here in this model 1, 13.26, of developing AKI. And this was really truly astounding that we could separate the development of AKI from a more stable hospital course in the setting of COVID-19 by plasma suPAR. And I don't have the paper here today, but there was a Nature Medicine paper published by another group from Greece on Unkinra treatment, stratified by suPAR level in critical care patients. I think it would be a very nice paper to also take a look at. We looked at the AKI a little bit more in this paper and injected an anti-suPAR antibody in our suPAR transgenic mice that we gave contrast dye to. And as you can see here, the suPAR transgenic mice that received an IgG control antibody or the suPAR antibody was a big difference in the development of AKI. So the ones that got the antibody still had some mild degree of renal failure, but it was much improved over the animals that got an IgG, suggesting that suPAR is in the causal pathway to AKI. And here you see that these suPAR transgenic mice under iohexol, which is the contrast, develop very strong ballooning of the tubules. And with the UPAR antibody, there's still some ballooning, but overall the kidney histology is much improved. And with that, we started to work in the pathogenesis of acute kidney injury on top of the work that we have done and others have done in the integrin field in the podocyte. We have a core together with the University of Michigan. Salim Hayek is the PI. It's called ISIC, International Study of Inflammation in COVID-19. We have, I think, more than 2,000 patients now that we follow since the beginning of the pandemic. We have serial suPAR and plasma and imaging modalities that we have charted. And what's really interesting is that hospitalized COVID patients who have heavy proteinuria had a medium suPAR level of 10.4 nanogram per milliliter, which is extremely high, and an incidence of AKI of 70%. Whereas hospitalized COVID patients with less than 3+ proteinuria or no proteinuria had a medium suPAR level of 6.7 nanogram per milliliter and an incidence of AKI of 31.4%. So what we learned from that is that if suPAR is high, there's a more higher chance for AKI. If it's extremely high, then the glomerulus gets damaged as well. And when one looks at the pathological finding under COVID-19 and the kidney, we really see that because we do see podocytopathies, immune-mediated glomerular diseases, but we also see this acute tubular injury, and we also see post-transplant kidney diseases. So we do see the full spectrum of kidney diseases. And it seems like the level of suPAR points us to, is it a one-component type kidney disease, or is it truly a multi-component, meaning glomerulus plus tubular kidney disease? This is important work that also showed, and I don't want to dismiss this, that the virus is sitting in kidney cells. And we don't fully understand what the contribution of the virus in kidney cells really is for the pathology. And I want to remind us, in the HIV field, we also saw HIV virus in kidney cells. And while we speculated and thought that the virus directly contributes to kidney disease under HIV, I would say today we have learned that that is not or has not been the major driver of kidney disease. It's been through the immunological activation that comes from HIV infection rather than by the virus itself. And I think, and it's a personal opinion, we have to wait for the data, that the virus, SARS-CoV-2 virus in the kidney is probably not so much the driver itself as the inflammation and the activation of innate immune mediators such as suPAR and perhaps others are. So as I told you, we do see a glomerular injury in very high suPAR settings. And that led to a term that we call COVAN, COVID-19 associated nephropathy or collapsing glomerulopathy. And in order to study that, we did another real interesting experiment. And I wanted to share one slide with you today that I'm happy to share with the public as we're finishing this study. We took our suPAR transgenic mice, and we took UPAR knockout mice that have no UPAR, no suPAR, and we took wild-type control mice, and we inhaled the human spike protein of SARS-CoV-2 every day through the nostril of these mice. And what is so interesting is, and we have some molecular detail how this works. This is not depicted here. This is here simply the mouse study. We can show that the mice that after 11 days that have received daily spike through the nose start to develop heavy proteinuria. So having high suPAR plus the spike seems to cause a reaction in the kidney that leads to a heavy proteinuria kidney disease, whereas the UPAR knockout mouse doesn't react to the spike that's blown in, so to speak, through the nose of these mice. And the wild type mouse that has a normal suPAR level also doesn't particularly react, or there's no reaction really on the kidney function with respect to spike coming in through the inhalation system. So with this, I conclude today that suPAR is an immune-derived circulating risk factor for kidney disease. SARS-CoV-2 is a strong inducer of suPAR, just like HIV and other viruses. And not every virus does this, by the way, but some do. Animal studies suggest suPAR to be a cause of kidney diseases. Clinical trials will be next to assess its potential as a therapeutic target. And with that, I'm excited to share this NIH-funded trial. Again, Salim Hayek from the University of Michigan, cardiologist. He's the PI. I'm a co-investigator. We just got funding for that. We're going to take a new suPAR removal device that was developed over the last years together with Miltenyi Biotech in Germany. So this is a specific device that only removes suPAR through this apheresis machine on this filter. And patients who have COVID-19 infection who need hospitalization with a suPAR above 6.5 nanogram, because that's the threshold for having a very high risk for acute kidney injury, they will undergo a selective suPAR apheresis treatment for 5 days, versus the control group will get a sham apheresis. And so we're very excited to modify suPAR, in this case in humans, and see what the outcome will be with respect to kidney injury. As you know, Jay Rappaport has enormous capability with non-human primates, has a kidney model that's being set up as well, and we're studying together with him also suPAR and other parts of the innate immune system, and hope to work with him on some other species as well. With that, I'll stop today and just want to show you, this is my laboratory. Cheng Li Wei, the gentleman right behind my left shoulder, really has been working with me on suPAR for the last 16, 17 years. This is Unzel Ham, who worked with me and David Skadden in Boston to find that the bone marrow immature cells are strong producing site for suPAR. Sanya Seva, my long-term collaborator at Harvard, and then Salim Hayek, who I've been working with the last 7 years, extremely successful work. We are about to publish a genetic paper showing that there are SNPs in the suPAR gene that are associated with certain outcomes. We're very excited about that. And with that, I'd like to thank the NIH, the NIDDK, and all the other institutes for very generous funding over the years that helped to push this work forward. And I want to thank you all again for giving me today time to talk about it. I wish I would have been able, probably in 2-3 months, I have some data on long COVID and suPAR level, but I would be very surprised. And again, this is speculative what I say right now. I would be very surprised if long COVID patients wouldn't have a more persistent elevation of suPAR level, but we'll have to see that. So with that, I'll close and I look forward to questions. Thank you.
Thanks very much, Jochen. That was a great talk. Can you answer the question for Kent Lloyd, which is on the Q&A? Based on these results, is the renal pathology seen in patients with COVID considered part of acute disease and or explainable by organ pathology, not really representative of PASC?
Yeah, that's a great question. So I think acute kidney injury, which is the main issue we see under acute COVID, this is something that happens under immunological attack of the kidney. And what we see there in terms of changes are very typical. Also, we would see in a patient that has no COVID and has, let's say, low blood pressure or sepsis. So there's, in a way, there's nothing special about it. It's just an accelerated storm on the kidney. Now, under PASC, what I do believe is we do see something like what we see with transplant glomerulopathy, where people have, after kidney transplantation, after 5, 10, 15 years out of transplantation, they have immunological activity and they have immunomodulation, and they get very specific changes in the pathology, like duplication of membranes and degenerative changes. And I think in PASC patients, we most likely will find more of those chronic changes that come from a persistent activation of inflammatory pathways, and that comes also perhaps from the virus being active within kidney cells as well. And that is something we need to study now that we have 10 months, a year and a half of post-COVID time. We need to study whether these patients who have chronic PASC, whether we do see in their kidneys these type of structures.
We have another question from Dr. Prabha Fernandez. What is the stimulant that causes suPAR production?
I'm sorry, can you repeat that for me, please?
Yeah. So from Prabha Fernandez, what is the stimulant that causes suPAR production? If one takes it, would be more made in COVID?
Yeah, so I think, and I hinted at that, it might be the spike itself that allows for a strong activation of innate immune cells. And I'm glad that question has been asked. I would need another 15 minutes to go a little bit into the concept, but I think the idea of the immune system to upregulate something like suPAR is to adjust the kidney for an infectious situation. So the kidney is tight for molecules that are larger than albumin. So everything that's larger than 60 kilodalton cannot be easily excreted from the kidney. Now, under viral attack or under bacterial attack, there are complexes of proteins that are larger than 60 kilodalton, and it's very hard to eliminate those from the body. But by sending something like suPAR to the kidney and opening the food processes, one would allow for a transient sort of like increase in permeability, and one can excrete larger molecules. It's somewhat similar to the idea of diarrhea, the idea of which is to debulk from infectious agents. So I think that might be the underlying idea why the immune system is linked to the kidney. And in order to do that, the innate immune cells are recognizing through pattern recognition, maybe through the spike with a specific interaction, to make more of these messengers, activate the kidney so that the kidney is ready to excrete larger molecules. Now, if that persists, then something that is a regulator becomes maladaptive and a chronic disease. That's the idea.
Another question I have is there any evidence that suPAR could be important in pathogenesis in COVID in other organs? For example, if it's produced in the lung in response to COVID, is there any effects in the lung or on blood-brain barrier or in the brain or in other organs? What is your thought on that beyond kidney?
Yes, so very important to understand is that CRP, for example, is a measure of acute inflammation. And there's a situation in which suPAR goes along with CRP. But there are many situations in which suPAR does not go along with acute inflammation, because suPAR is more a marker of a chronic inflammatory process. And so what we have seen in publications over the last year and a half is that suPAR levels are very good predictors for acute lung injury in COVID and also for longer term COVID lung outcomes. It has to be studied more in neurological diseases. There are some studies before COVID that link higher suPAR level to neurological diseases, but the information is much more scarce. But there is good literature already on acute suPAR level, the levels thereof, and acute lung injury. What the mechanisms are, I can't tell you. I think it's striking that suPAR expression is high in the bronchial tree, and the lung is an organ that is massively attacked by COVID. I think that makes sense. And I also can understand why the bronchial tree cells, the stem-like cells, have a system in place to perhaps sense air that's coming in and react to it in an innate immune fashion. But how this works is really not very well understood. The pathological steps of how suPAR and uPAR work in the kidney is relatively well worked out over the last 10, 15 years. And in other organs, it's at the beginning. I can tell you in the suPAR transgenic mice, if we don't do anything to these mice and just let them sit for a year, the finding we have is kidney disease and not lung disease and not heart disease. If we do additional things to them, then we can also trigger additional organ dysfunctions. But the prime target of suPAR seems to be the kidney.
We have another question from Ryan Carner. Is there a role for platelets in elevation of suPAR levels?
I don't know the answer to this. I think that's a great question that I haven't fully thought about it. Would the idea be that if there's depletion of platelets, that this could particularly lead to activation of the innate immune system? Or whoever asked the question, could he or she just kindly elaborate a little bit more so that I can connect the thought a little bit better? Or was it just because we see platelet dysfunction in active SARS?
Yeah, well, let's go on to another question. Yeah, go ahead. Ryan, are you on? Okay. So Prabha Fernandez also asked, would the spike protein without the RBD domain also activate suPAR?
We don't know. What we do know is the RBD domain does not. But the spike without the RBD, I don't think we have tested that yet.
Okay.
And there's a lot that we need to learn. But if you take, for example, hantavirus infection, hantavirus enters the cell via beta-3 integrins, and the suPAR levels facilitate entry of hantavirus. And so we think that suPAR and the spike of SARS have a particular interaction at the integrin, which facilitates the phenotype development. And there's a particular role for the spike here, as in contrast to the RBD domain. But whether the spike needs the RBD domain or not, I don't think we have tested that yet, but we will. That's a great suggestion and question. Thank you for that.
Okay. So, Dr. Reiser, I want to echo the comments of Dr. Fessel in the chat. I want to thank you so much for an outstanding presentation, and thanks also to the audience for their attendance, and also for a great discussion. And this has been a really, truly great start for our series in this year's 2022 round of talks. So thank you so much.
Thank you all so much, and have a happy and healthy 2022.
You also. Thank you.